Okay, so welcome back to this next video in which we are discussing ion channels and cancer. Okay, right, so we're currently in the process of looking at specific examples of ion channels that are involved in cancer. Okay, so in the previous video what we saw is the Agogo-1 channel, which was made up of 4 Agogo-1 subunits, okay, and which was ectopically expressed in around 75% of cancers. Okay, what we now want to turn our attention to is another Agogo channel, which is the Agogo-2 channel, okay, which is made up of 4 Agogo-2 subunits. Now, the Agogo-2 subunits have another name. They're also known as KV10.2 subunits, just like Agogo-1 subunits were also known as KV10.1 subunits. Okay, right, so the Agogo-2 channel then is a homotetramer of 4 KV10.2 subunits, like so. So here are these 4 subunits bound together to make a homotetramer here. And this is a voltage-gated potassium channel that we're forming here. Right, okay, so the Agogo-2 channel seems to be extremely important in the cell cycle, and it seems to be extremely important in a phenomenon known as premitotic condensation. So let me explain to you what premitotic condensation is. So basically, prior to the mitosis phase of the cell cycle, what happens is cells hugely reduce their volume, okay, within their cytoplasm, and this causes them to condense down until they are a beautiful sphere. So let me show this. So normally a cell might have some really bizarre shape, especially in an epithelial cell, which is generally the cells in which cancer arises. Okay, so here this might be our cell originally. However, prior to the mitosis phase of the cell cycle, what happens is it condenses down, so it hugely reduces its volume and it becomes spherical. Okay, and there's another name for this. It's also known as mitotic rounding, okay, referring to the fact that the cells become much rounder prior to mitosis, or rather during mitosis. Okay, so this is the phenomenon of premitotic condensation, which is sometimes abbreviated down to PMC for short. Okay, so let's have a look at which specific portion then of the cell cycle premitotic condensation actually occurs in. So let's redraw out the cell cycle. So remember, the cell cycle firstly consists of this G1 phase, which I'll show here. Okay, so I might even make it a little sort of a pie chart like this. So here's G1 phase. Okay, then you've got the S phase here. Okay, and remember, in G1 phase, the cell begins to duplicate proteins, getting ready for division. In addition, it also starts to prepare the apparatus that you'll need to copy the DNA. So it gets ready for synthesis phase, S phase here, in which all 46 chromosomes are copied. Okay, and then the next phases of the cell cycle then are the G2 phase and the M phase. Okay, so this is the second growth phase here, G2, and then here is the M phase of the cell cycle. Right, okay, so premitotic condensation occurs right at the end of G2 phase before you then go into M phase. Okay, so cells start to shrink their cell volume towards the end of G2 phase and then at the beginning of the M phase. Okay, now this goes on throughout the first few portions of M phase. Okay, particularly it occurs in prophase, prometaphase, and then gets to the top portion. It gets to its finest moment at metaphase, and then it starts to reverse in anaphase, telophase, and cytokinesis. Okay, so let's just talk about the stages of M phase of the cell cycle. Okay, so the first stage within M phase is what's known as prophase. Okay, so stage 1 is prophase, and in prophase what begins to happen is the chromatin condenses into the actual chromosomes, so the chromosomes become visible. So let's show this. Okay, so let's say we're going from this original cell here. Okay, so the chromosomes are going to become visible, so I might start by drawing the nucleus here. And what's going to happen is the sister chromatids attached to one another are going to become visible. Okay, and that's a phenomenon known as chromosomal condensation. Okay, when the chromosomes condense down. So this is chromosomal condensation. Okay, right. Then the other major thing that occurs in prophase, and I'll just highlight this up. So chromosomal condensation is shown here by those sister chromatids now being visible here. Okay. The other major thing that occurs in prophase is that the mitotic spindles start to assemble. So you also get mitotic spindle assembly. Okay, now a mitotic spindle is a special cytoskeletal structure that's constructed out of microtubules that is going to be involved in splitting the sister chromatids apart. So mitotic spindle assembly. Okay, so I'll show these here. So you have two of them, one at either pole of the cell. So here's one of them with the centrosome at the center, and then microtubules which are attached to the centrosome coming off towards the center of the cell. And then here is another mitotic spindle here. Okay, so again I'll color code this in red. Now in prophase what will start to be happening is this pre-mitotic condensation that we're studying here. Okay, so the cell will start to get smaller. Okay, so I'll show this gradually occurring as we go through the stages of M phase. So here I hope I've slightly shown it decreasing in cell volume. It's debatable as to whether I have, in the ideal world, that drawing would be slightly smaller than this one. Okay, right, and it's going to continue in the next phase of M phase, so let's go through the second phase of M phase. So in the second phase of M phase, which is known as prometaphase, what's going to happen is the nuclear envelope is going to break down and you're going to get what's known as chromosomal capture. Okay, right, and of course we're also going to be getting the pre-mitotic condensation, so I might draw this first this time so that I'm bounded. Okay, so here now the cell volume, hopefully you can see, has hugely decreased. Okay, so what's going to happen is the nuclear envelope is going to break down, so now the sister chromatids are going to be on their own. Okay, and you're going to start getting chromosomal capture, so microtubules are going to start connecting to the sister chromatids. Okay, so I've only shown 2 sister chromatids, but of course there would in reality be 46 sister chromatids. Okay, so let's put the 2 key things here that occur. So nuclear envelope degeneration, okay, nuclear envelope breakdown, or I might just put nuclear envelope goes cross, okay, and now the sister chromatids, which I'll highlight in blue here, are now on their own in the cytoplasm. Okay, so that's one of the key things that occurs in prometaphase. And then the other thing is that some of these microtubules of the mitotic spindles are going to actually attach to the sister chromatids. Okay, and this is a phenomenon known as chromosomal capture. Now it's not going to be complete yet. It won't be complete until every single one of the sister chromatids has a microtubule from both mitotic spindles attaching to one of the two in the pair. Okay, so each sister chromatid is a pair of identical sister chromatids. Okay, so basically what needs to happen is a microtubule needs to, from this mitotic spindle, needs to attach to one of the pair, and a microtubule from this mitotic spindle needs to attach to the other of the pair. Okay, right. So here is the phenomenon of chromosomal capture here, where these microtubules are attaching to the sister chromatids. Okay, so that's occurring in prometaphase, and the thing that we're seeing here is that the cell volume is going down. Okay, so pre-mitotic condensation is occurring through the second phase of M phase, and then it's going to reach its finest hour in the third phase of M phase. And the third phase of mitotic phase is called metaphase. Okay, and by the time metaphase occurs, the cell will be beautifully spherical, like so. And what will now happen is the sister chromatids will be lined up right down the middle here. Okay, all 46 of them. Okay, and the mitotic spindles will have attached up to them. Okay, so every single sister chromatid will have a mitotic spindle microtubule attached to it, And the idea is that the sister chromatid pairs are going to be attached to opposite mitotic spindles. Okay, so this is what's happening in metaphase. Here is the mitotic spindle microtubules here. Okay, and here in turquoise, here are our sister chromatids, and I'm only showing 3, but it should of course be 46. Okay, right, so that's what happens in metaphase, and we've reached the peak of pre-mitotic condensation now. The cell is beautifully round, we've got beautiful mitotic rounding, and now as we progress through M phase further, the volume is going to go back up now, it's going to reverse. Okay, so through anaphase, which is the next phase, you're now going to get bigger. Okay, now in anaphase, what's going to occur is the mitotic spindles are going to contract basically and pull the sister chromatid pairs apart, and they're going to end up pulling one of each sister chromatid to the opposite sides of the cell. Okay, so let's have a look at this, and of course the cell's also going to increase in volume, so it's going to go like this now. Okay, so like so. So here is one mitotic spindle, here is the other mitotic spindle, and now they've got one copy of each sister chromatid attached to them like so. Okay, so again, here are the mitotic spindles in red. Okay, here in turquoise then is one copy of each chromosome basically now going to each side of the cell. Okay, then in the 5th phase of M phase, what's going to occur is you're going to get the nuclear envelopes reforming. So telophase is the 5th phase of the M phase. Okay, so the nuclear envelopes are now going to reform, and you're going to begin the process of cytokinesis. So what's going to happen is the cell is going to adopt this sort of dumbbell shape like so. You're going to reform nuclear envelopes in the 2 separate portions here, and each one of these new nuclei is now going to have a copy of all 46 chromosomes within it. Okay, so here are the chromosomes, and we've reformed the nuclear envelopes now. Okay, and the volume is going up still. Okay, and then finally the 6th phase of M phase, which is not part of mitosis, even though it is part of M phase. So this is important to understand. Mitosis, the process of mitosis, just means the division of the nucleus into 2 identical nuclei. That's occurred now. 1, 2, 3, 4, and 5. Those have completed mitosis. Okay, this final 6th phase of M phase is counted as part of the M phase, or the mitotic phase of the cell cycle, but it's not, strictly speaking, part of mitosis. Okay, so the 6th and final phase of M phase is what's known as cytokinesis. Okay, and this is where the cell actually does split into 2 separate cells. So like so, here we have now the 2 separate cells with their nuclei in them. Okay, and each one of these cells will have a nucleus that contains 1 copy of all 46 chromosomes. Okay, and that's the cell cycle finished. Okay, right, so that's the phenomenon of pre-mitotic condensation and the role it plays in this M phase of the cell cycle. Now what we want to look at is how does this relate to ion channels, and specifically, how does eferagogo-2 play a role in this? Well, basically, eferagogo-2 is not usually expressed at the plasma membrane. Okay, usually, if I draw a cell here, usually it appears if you do immunocytochemistry. Okay, if you try and find where it is in the cell using immunocytochemistry, what you find is that the EAG2 channels, they usually appear in the cytoplasm. Now, they're probably not just free in the cytoplasm, they're probably stored in cytoplasmic organelles known as endosomes. Okay, so an endosome is a very large intracellular organelle that's membrane bound. And basically, when you want to take membrane proteins out of the membrane and store them inside for a while, potentially because the cell wants to desensitize to something, they're often taken to the endosome temporarily. And it seems that for EAG2 channels are usually stored in the endosome rather than actually being on the plasma membrane. Okay, so here is the EAG2 channel in the endosome here. Okay, very little of it is usually actually at the plasma membrane. Now, basically, what seems to happen is in the G2 portion of the cell cycle, towards the end of G2, what happens is the EAG2, the efferogo2 channel, is moved from the endosome to the plasma membrane, and this is what causes the reduction in cell volume. Okay, so what's going to happen is pre-mitotic condensation is caused by moving EAG2 from the endosome into the cell membrane. Okay, like so. Now, why does this cause pre-mitotic condensation? Well, basically this alters cell volume homeostasis in the cells. Now let me explain how. Basically, when you move lots of EAG2 into the plasma membrane, it's going to allow potassium ions to move out of the cell. Okay, now what then happens is another ion follows it for a different channel. Okay, so chloride anions are usually passively distributed across a cell membrane. So what's going to happen is when you move these potassium ions out, because you're moving positive charge across the cell membrane, you need to counterbalance that, and what's going to end up happening is chloride anions are going to move from the cytoplasm into the extracellular space through another channel. Okay, so understand that chloride anions are usually passively distributed. Okay, so they can just move like this to follow charge, basically. Okay, so this channel then that's going to allow the chloride anions to leave the cytoplasm of the cell and go into the extracellular space, this is believed to be the CLC3 channel. Okay, which is normally at the plasma membrane. So you might wonder, well, why is it not normally just letting the chloride leave? As I say, chloride is normally passively distributed. Okay, so it's normally at an equilibrium whereby the amount moving in and out through this channel will be the same, basically. Okay, but when you move this EAG2 channel into the plasma membrane, now of course potassium is going to start moving out. The chloride will now start having a net movement out to counterbalance the potassium ions that are moving out. So now you will net move potassium chloride out of the cell. Okay, so you're moving salt out of the cell, and of course that's going to make the extracellular fluid hyperosmolar and the intracellular fluid hypoosmolar. Okay, and that osmotic gradient is then going to cause water to move out of the cell as well, and the water will move through special water transporters known as aquaporins that are in the plasma membrane. So this here, this is representing an aquaporin. Okay, and water molecules will move through via the aquaporins, I should say, rather than through, okay, to cross the cell membrane. And that then is what causes the reduction in cell volume. Okay, so this is happening from the end of G2 all the way through prophase, all the way through prometaphase, all the way through metaphase, and then at the end of metaphase it seems to reverse. You bring the EAG2 channels back from the plasma membrane and store them back in the endosome. You reverse this, okay, and that then returns the cell, as far as the ion channel composition on the membrane is concerned, back to what it was before, and then the equilibrium is re-established. Okay, so the cell eventually goes back to having the cell volume that it originally had, because that was the cell volume that it was at equilibrium with before. Okay, so when it had its normal complement of proteins in the membrane that were all moving ions and water, okay, the equilibrium volume that it had was that previous equilibrium volume. We distorted the cell volume homeostasis mechanisms by adding this new ion channel into the plasma membrane. That meant that the new equilibrium volume was much lower, okay, because of the fact that it was allowing salt to leave the cell, okay. But now we've removed it, you'll return back to the equilibrium that you had before, so the cell volume will gradually build back up as that equilibrium is rebuilt. Okay, right. So that's believed to underlie this pre-mitotic condensation that occurs prior to the M phase and during the M phase within the cell cycle. Okay, and that's very, very, very important for cell cycle progression that this actually occurs. Okay, right. Now, what was actually found was that EAG2 expression is far too high in certain cancer cells, okay, particularly cancer cells of a type of cancer known as medulloblastoma. So let me tell you about what a medulloblastoma is. Okay, so a medulloblastoma tumour is a tumour of the brain, and it truly, we think, is a tumour of brain cells. Tumours of brain cells are quite rare. Generally, if you get a tumour in the brain, it's generally not actually of neurons, because neurons have such low ability to divide. But a medulloblastoma does seem to be a tumour of brain cells. So let me describe where it is present. So if we look at a brain from the left-hand side, if I draw a brain from the left-hand side, here is the left cerebral hemisphere viewed from the side here. And then poking out from underneath it, you'll have the brain stem structures, like so. So let me colour in these different portions. So here, this in blue, this is the spinal cord. Okay, so that's the spinal cord. Then, as the spinal cord gets to the top, it will go through the great hole at the base of the skull, known as the foramen magnum. So this ring here around the spinal cord, that's the foramen magnum. That's representing the hole at the bottom of the skull, a very large hole at the bottom of the skull. And I've got that the wrong way around, magnum. Okay. Then, above the spinal cord, the continuation of the spinal cord effectively is the brainstem, and the two portions of the brainstem that we can see here are the medulla sitting on top there. Okay, that's the immediate continuation of the spinal cord, and then above the medulla in turquoise here you then have the pons. Okay, now behind the brainstem, sitting underneath the cerebral hemisphere here, you then have the cerebellum. Okay, which literally means little brain. Okay, now a medulloblastoma is a tumour that occurs, or is believed to occur, from cells of the cerebellum. Okay, and what happens is generally it's at the front of the cerebellum and therefore generally protrudes into the back of the medulla here. Okay, so generally you get a massive great lump in this sort of position here. So that's what a medulloblastoma is. It's a massive great lump that's protruding from the cerebellum forwards into the back of the medulla, okay, and also into the back of the fourth ventricle, because there is a cavity here containing cerebrospinal fluid on the back of the brainstem behind the pons and the medulla that's called the fourth ventricle, and that will also get crushed by this growing mass. Okay, so that's our medulloblastoma there. Okay, and in these cells, one of the utter hallmarks of medulloblastoma cells is that you have a very high expression of ether agogo 2. So ether agogo 2 levels are really aberrantly high in medulloblastoma cells. Okay, so given what we now know about how important ether agogo 2 is for the mitotic phase of the cell cycle, we might hypothesize that there might be something in this. Maybe ether agogo 2 is a driver, okay, that drives the overproliferation of these cells. Okay, now basically it hasn't really worked, okay, because what you can do is you can do the same thing as we did for ether agogo 1. Okay, in ether agogo 1 we expressed it in a heterologous system, and that caused the malignant transformation. Overexpressing ether agogo 2 in heterologous systems, i.e. in cells that haven't normally got it expressed, okay, does not cause malignant transformation. In fact, it causes the exact opposite. The cells do not divide, instead they commit apoptosis, they commit suicide. Now, why is this? Well, basically, if you take a heterologous system, some cell that doesn't normally express ether agogo 2, and then suddenly force it to express e-for-a-go-go 2, what ends up happening is it ends up with e-for-a-go-go 2 channels all over it, okay, all over its plasma membrane. Okay, it doesn't basically restrict them to the endosome except at the pre-mitotic condensation period. Okay, it ends up with them all over the plasma membrane, and what this leads to is the cell being permanently shrunk. Okay, the cell ends up tiny and it ends up committing apoptosis, and it's certainly not capable of over-dividing. Okay, so what that tells us is something must orchestrate this movement of the e-for-a-go-go 2 from the endosome to the plasma membrane, and make sure that it only goes to the plasma membrane at that exact period where it's needed there during the M phase. Okay, and we don't know what that currently is. But e-for-a-go-go 2, maybe it's not a driver, maybe it is just a consequence there. Okay, so it's a consequence of some other driver that's then driving its behaviour because e-for-a-go-go 2 is so essential for the division process. Okay, but it's still an interesting thing to note that these medulloblastoma tumour cells have very, very aberrantly high expression of e-for-a-go-go 2. Okay, right, so that's e-for-a-go-go 2. We'll call it there for this video, and in the next video we'll start to look at examples of ion channels which seem to perform their role in carcinogenesis via having effects on calcium signals, which then seem to drive cellular proliferation.